Okay, so as we start to hit into the third macromolecule that we're lecturing on, proteins, okay? So, what we need to know about proteins is what I have already said before. Proteins are some of the most diverse and complicated in the body, and some of the most important that we have to go through. Okay, so why are they so important? Well, actually, they take 50%, more than 50% of dry mass of cells in a protein. So proteins are polymers containing repeating monomers, which is those amino acids. So remember I said amino acids were the building block of proteins. So you have many different amino acids coming together to make a polymer, and the polymer is a protein. Okay? So, going on to the second one, proteins are essential for cellular membranes. Remember, you have those phospholipid bilayers. Now, proteins are extremely important in the cellular membranes because it dictates what can go in and out of the cell. Oxygen carrying pigment, that's another question that you had actually on one of your CK12 is hemoglobin. Is a protein. So if you are anemic, and often that means that your hemoglobin is quite low. Often you feel tired, fatigued, because your hemoglobin is essential for carrying iron, which carries oxygen. Okay, so the iron that gives us our color and our pigment and what that looks like. Also, proteins are antibodies. So they're part of your immune system. So they are your first line of defense when you have a pathogen that comes, or a foreign object that comes into your body, and your body's like, you don't belong here. We don't recognize you. You look like you could cause some damage. Your immune system kicks in, and your antibodies are released so that it can tag the pathogen, and then the white blood cell can come in, eat it, and then take it and try to get rid of it and kill it off. Okay, so also moving on, hair and surface layers of the skin. Your integumentary system, which is a part of your skin, is your largest organ system in your body, actually. Your hair that you have, and you also have like fuzz hair. Everybody has it. Little tiny ones, if you can see them or not see them, are on the surface layer, and that actually has a protein called keratin in it. Also, collagen is another protein, and collagen is actually found in many different tissues in your bone, your tendons, and your ligaments. So you can have muscle to bone or bone to bone, which is your ligaments, that help. Muscle that extends into the bone is your tendon. So that is all collagen, and that is actually a protein. So when we say that protein is one of the most diversified, it absolutely is. One of the things you need to know about the building blocks of proteins, amino acids, these come together to make up a protein. There are 20 different amino acids found in living organisms. What is interesting about that is 11 amino acids your body naturally makes. But 9 of those amino acids you need to take in through your nutrition. If it's through supplements, through plant-based, that has to do. So if you are not getting enough of a certain nutritional or a well-balanced diet, maybe you're not taking in an amino acid that you essentially need for your body to function at full capacity. So once again, structure of amino acid, what is it made of? Well, it's made up of one amino group, which is an NH2, a carboxyl group, which is HOOH, and then a variable group. So we will go through. So go ahead and go on to, which would be slide 4. This is the general formula of an amino acid. So you have your amino group, and then on the opposite side, you have your carboxyl group, which is that COOH, and then you have the variable group. And remember that variable group will change all the time depending on what attaches will determine the structure of that protein. Okay, so let's move on. All right, there are different amino acid groups, or classes. So you have the generic one, which is the basic formula, which is your amino acid, your variable group, and your carboxyl acid group. Okay, so that's in the center. When you start adding the 20 different amino acids to it, remember, that goes in one of those like alanine, aspartic acid, cysteine, or histidine. So there's 20 of them. They will actually be, one of them will plug into the variable group. And when that happens is that variable group will determine what its structure is, and structure determines a job, what it's going to do. And so that is, so basically it's function. So for proteins, amino acids, the variable group, structure determines function. So this will make more sense as we go through on how proteins are attached to each other. So I'm not going to spend a whole lot of time on slide 6. Just know that you have 2 amino acids that will come together, something smaller to form into something bigger. Remember, the water molecule is taken out through condensation reaction, and then a peptide bond is formed, and then you have a larger molecule. All right, protein structure. So you have one of these 20 amino acids that come together, and they're all like lined up. So what do structures of a protein looks like? So you have 4 different ones. You have primary, secondary, tertiary, and quaternary. And quaternary is when you have many different that are coming together, and it will make sense a little bit later. So primary is just a chain of amino acids. Secondary, if you go back to the primary, you could see that condensation reactions have happened to allow these amino acids to start attaching to each other. So then it gets a little more complicated when you go up into secondary structure, and you have 2 structures within the secondary structure. You have an alpha helix, which is a spiral again. Remember, we saw that before. And then a beta pleated sheet, which actually looks like just a folded sheet coming up and then crunched out. And so you have alpha helix, which plays a really important role in our DNA, and then beta pleated sheets as well. So remember, these are all bonded when we get into this point by hydrogen bonds. All right, you start to see what an alpha helix looks like. You don't need to memorize that on your slide 10. Just make sure that you understand it's a spiral, and then it sits on that spiral as we start to look at that. All right, continued on to slide 11. It's still the same thing. You can just look at that. On slide 12, when we're talking about an alpha helix, the R groups, those variables that start to the 20 amino acids that we had talked about, the different kinds, they start to stick out. And because the variable groups stick out and are not the backbone, but they're kind of sticking out, they actually allow reactions and then allow it to go and bond with others to create a more complicated protein, and skip to the next level of the tertiary or quaternary structure of a protein. So going back to slide 13, the beta pleated sheets, you can start to see they're just kind of folded sheets that kind of lay out. And then that's all we're going to know about that. So when we talk about complicated and how those variable groups stick out to allow bonding to a next level of structure, let's look about tertiary structure. It is folded like a snake. We are on slide 15. So it's green. You can see it's really slided, but then you start to see an alpha structure being created in there. And what allows that snake-like folding up in there is, one, those R groups stick out to allow the bonding, but hydrogen bonds. And those hydrogen bonds, along with disulfide bonds, ionic bonds, and van der Waals forces. And van der Waals forces means that it's almost like a magnet. So folding happens, but how does it stick and stay there so tightly bonded that the structure doesn't change? Well, it's kind of like a magnet. You get a negative and you positive, and you get them just close enough, and what happens? They suck together and they're pretty tightly bonded. So that's van der Waals forces. It's not quite a bond, but it's almost like a positive and negative magnetic force that allows it to stick and stay structured very tightly together. And so it allows the protein to stay that way. Which then, after you get to a quaternary structure, a tertiary structure, which now is folded pretty tight, you can actually go to a quaternary structure. And what does that mean? It means that you have a combination of tertiary structures, which are either made up of alpha structure or a beta pleta sheet, and those come together and they bond with the tertiary structure, and then a tertiary structure bonds with another tertiary structure until you get a really complicated protein like hemoglobin. And hemoglobin is a quaternary structure, and because it has iron in it and other things that it's carried. And so you really are coming together with the same thing. How do a tertiary structure stick with another tertiary structure to another one? Well, it's because of hydrogen bonds, disulfide bonds, ionic bonds, and van der Waals bonds. So it's kind of like that magnetic force. It's the right combination, makes it stick really good together. Okay? So what happens when you have different kinds of quaternary structure, and they're vary? Well, you have two different types of proteins at this point. You have a globular and a fibrous protein. And proteins are divided into these two really broad group. So it really depends on what type of structure is it. If it's a primary, secondary, tertiary, or quaternary structure. And that's what we're looking at. Slide 18. So what you will see the difference on slide 18 between a fibrous and a globular structure. So a globular protein are large. They have hydrophilic R groups. That means that you have R groups, those amino acids, the 20 different ones which go into those R groups are going to like water. They're soluble in water. And so those are enzymes and plasma proteins. And then you have fibrous proteins. These are long ropes. So what are long ropes? They're like this. They kind of spiral out. And actually, one of the big differences between globular and fibrous proteins is globular is hydrophilic, but fibrous is hydrophobic R groups. That means they do not like water whatsoever. They do not like being exposed to the outside. And these kind of things are collagen, which we talked about collagen before, keratin in the hair. Remember, collagen is found in your bones, tendons, ligaments. Elastin, which that's flexibility within the skin. And then actin and myosin. And actually, actin and myosin are proteins that are found in your muscles that allow the contraction. And it's almost like a walk-like contraction, and then it releases out. So actin and myosin are for contraction purposes within your muscles. All right, and 19 and 20 are actually pictures between the globular protein and the fibrous protein. Globular protein, remember, are hydrophilic, and so they are 90% of those globular proteins are enzymes, And enzymes are used to create chemical reactions within your body with as little energy as possible. And then you could see on there's a really pretty picture of that globular. And then the fibrous protein is really long rope, and you could see actin and myosin that I have a picture. And I'll actually post a really cool short little video of how actin and myosin actually work in the muscles. It's very cool to see it walk across, attach, and that's when your muscles are contracting, and then letting go, and it kind of walks its way back. So that's actin and myosin on proteins. Once again, if you have any questions, please join Zoom. If you need to have an appointment before or after, please let me know and we'll set up a time, okay? All right. Have a good day.